Susie, what do you know about the evolution of smell?
Absolutely nothing.
Nothing.
No, I don't know anything about smell. I've never.
What about your dinosaurs? Don't you ever stop to think about your dinosaurs smelling things?
Well, that's true. And you can look at kind of endocasts, like the cast
Yeah.
of the brain case,
Yeah.
to try and see whether, they've got big olfactory lobes or small olfactory lobes to kind of, maybe get a guiding idea. But, my dinosaurs are herbivores. I don't know, you can barely tell the olfactory lobes. I don't know anything about it. So, Rob, what is your least favorite smell?
Well, I can tell you that pretty clearly and specifically. That is when I used to research hagfish, I used to do these experiments. For a paper I published a while ago, we had to rot some hagfish for a taphonomy experiment. And hagfish are already disgusting when they're alive. So these blind slime monsters, jawless fish, and after they've been rotting and you get them out of the water, oh my God, I've never smelled anything like it. Made me physically heave. But hey ho, we got a paper out of it and it was quite nice. How about you?
That is disgusting. I think animals decaying is a particularly unpleasant smell. One of the most unpleasant things I've ever smelled is I joined a couple of dissections of crocodiles when I was studying legs and leg muscles. And I was just interested in the legs, I didn't need to see all the disgusting, gooey, soft parts. But this particular crocodile, it had its tail shut, like accidentally kind of slammed in the freezer door, and for some reason this had damaged the muscle tissue of its tail, and the tail had started to rot. And the person I was kind of watching this because I'm a geologist, I don't know much about dissection, and the person who I was doing the dissection with kind of sliced open the tail, and all this sort of sloppy, decayed muscle just out. It was absolutely disgusting. But I learned that there in the vet lab where we were doing it at the Royal Vet College, they put Vaseline under their nose so that all you can smell, or not Vaseline, and Vicks, Vicks vapor rub. So it's just so all you can smell is the Vicks vapor rub instead of the like decaying flesh of the disgusting animals. It was minging, and it reminded me why I prefer rocks to living animals.
Well, that's also a reminder about why smell is important, I guess. You need that smell in order to understand why things should not be done and avoided. And that's what we're going to find about in today's episode.
So, Rob, what have you been doing since we last met? We haven't seen each other for ages.
It's been a while, a long gap in between the two recordings. I have been off to Mallorca with the students for the first time. Yeah, for the first time, me leading a undergraduate field course. It was a lot of fun. We had fun.
That's very exciting. And it's a biology course, is it you're leading?
It's ecology and paleontology. We're doing both at the same time. There was more ecologists than paleontologists on there, but for the ecology side, we saw wild tortoises. We saw cuttlefish in the wild doing their little camouflage thing. We went to a vulture sanctuary. For paleontology, we were looking at all sorts of fun things. I found some Jurassic stuff, but also we were looking at Pleistocene stuff. So where this weird Myotragus thing is playing around. And the students found all sorts of weird fossils, including a fossil cuttlefish, which I'd never found before, which was kind of cool. So we had fun.
Wow.
And no one died. That's the important point. That was what I was worried about. Yeah.
This is a major thing on undergraduate field courses. I've very rarely taught field courses where just, everyone's fine. You come back and everything's fine. Most of the time that doesn't happen.
I don't think we even had an injury, which I'm relieved about. At one point, we took them out on a rocky shore at one point on a windy day, and I said we had to give up and say, no, I'm not happy with this. As they stood next to these enormous waves. Get indoors now. It was mostly sunny though. Anyway, I want to talk to you about mammals today. So our paper today is... Oh, no, hang on, stop. I want to do an update first. Fossil Files update section. So in one of our most recent episodes, we talked about the Ediacaran and the Cambrian, didn't we? About the evolution of animals in the Cambrian. So this was really interesting. So this is a very short update because I want to move on to talking about mammals. But this was the episode where we talked about how the Burgess Shale and the Cambrian is this explosion of animal diversity, but actually a couple of papers we pointed out eked into the example that we're probably going to find some bona fide animals preserved in the Cambrian. And then do you know what happened? About 2-3 weeks after we did that, an amazing paper dropped in Science from a Chinese team in collaboration with the University of Oxford. Did you see this paper, Susie? You're nodding your head in recognition of this.
I did. We had a little chat about it on Blue Sky, I think, at the time with
Matthew
Cobb as well. Hi, Matthew.
Yeah. Oh, yeah. Hey, Matthew. Big fan. So, yeah, this team from China with the group from Oxford, they found this deposit in the late Ediacaran in China, and it had this Burgess Shale type preservation, and they found a whole host of weird-looking animals and, fairly uncontrovertible animals in this non-bilaterians and bilaterians. So jellyfish, ctenophores, worms, things with guts, and having a gut is important. So there we go. Our prediction. I mean, obviously, it's not because of what we said. But the point is that the fossil files predicted and the fossil files delivered. Yeah, we saw it happening.
I thought you might actually do the paper, Rob, that came out as another one. But yeah, we're gonna move forward in time.
We are going to move forward in time, but I thought, well, we can't do that paper because we just did that exact topic. But then again, I've just realised that we're moving on to a very similar topic we talked about last time, because now we're picking up on the mammoths. In my most recent choice, we did mammoths, and I'm doing mammals again and smell again. So those mammoths that were unable to smell because they had allergies in that absolutely preposterous paper.
The mammoth sex. Mammoths died out because they stopped having sex because they had so many allergies they couldn't smell each other anymore. That was the mammoth extinction fossil fail.
Yeah, shall we swing in a different direction and ask about how we could pick up the evolution of smell from fossils and genomes in a more rigorous way, perhaps, than the mammoth example?
I do think this paper is more rigorous than the mammoth smell allergy sex paper. There's no photos taken through the glass of random.
No. But before we start giving the authors of this week's paper, an ego problem, that's a low bar to clear. To be better than that mammoth paper is not hard. Okay, so this paper is The Olfactory Bulb Endocast as a Proxy for Mammalian Olfaction, published in PNAS just at the end of last year. So it's a bit of an old one for fossil files. Sorry, everybody. This is by Quentin Martinez and a whole bunch of other people. So it's a combination of zoologists and paleontologists, mostly from continental Europe. In fact, I think almost all of them from continental Europe. So this is Germany and France in the most part, I think, most of these people.
Several Natural History Museum or former Natural History Museum members on this as well.
Some of your colleagues at the Natural History Museum do pop up on here too. Yes, absolutely. Great. So let's have a think about this. The pretext of this paper is that the evolution of smell is very important for mammals. I don't know why we're focusing on mammals particularly. I guess the evolution of smell is very important, but mammals is a really important part of this because they have lots of genes and it's an important part of the mammal story. And you want to construct the evolution of these things from fossils is quite difficult. So you need to think about that carefully, which is why I wanted to ask you that question about dinosaurs, I guess. So let's think about how we might do this, first of all. So they focus a lot on the morphology of certain parts of the endocast. So the endocast is the space inside the skull left behind by the brain in this case, right? So that's something you can fossilize. Now, brains don't fossilize, well, apart from very rare circumstances, which is an interesting story for another time, perhaps.
Do we know a fossilized brain?
Well, I could do a whole episode on fossilized brains, but that's a bit difficult because I have some academic skin in the game on that one. But there's lots of human fossilized brains which are interesting, and there are some fossil animal brains as well, dating back to the Cambrian and more recently. But they are rare. You have to have very specific circumstances to get a fossil brain. But the endocasts are much better because this is, you can use the hard tissues of the skull to get a handle on it. And sometimes you can get your endocast preserved in such a way that you can see the shape of the brain and the different subunits of the brain, and the olfactory bulb is a sort of bit that sticks out a little bit. So you can normally detect it roughly just looking at the skull if it's preserved in just enough detail, perhaps. Though we're going to come to this later on. Now, this reminds me, when I was a kid, I had a big book of dinosaurs. What were they called? Dorling Kindersley's, whatever, the DK books. Yeah, and it had a stegosaur in it, like a little dissection of a stegosaur. And it talked about the stegosaurs having a very small brain and then being very stupid. And it also said it has a secondary brain in its tail. And I remember that quite vividly as a kid. This is presumably made up, right?
Well, actually, stegosaurs do have very small brains. And we can tell this because they have the endocast that you've been talking about. We can reconstruct through CT scanning or whatever, but they actually have a brain case, which is a bony structure that sits inside the skull. And so we can see that their brains are, typically they were always quoted as saying about the size of a walnut. I believe my colleagues actually, with our very complete stegosaur Sophie, they actually calculated how big the brain was, and they said it's more like a plum. But that's very small because if you think about Stegosaurus, it's about 5 meters long and it's got the brain the size of a plum. Somebody once said, I think, that it had the smallest brain of any terrestrial organism, any terrestrial vertebrate that's ever lived, which does sound totally plausible. But I mean, that we know that brain size doesn't strictly correlate with intelligence, and that stegosaurs lived for a long time, millions and millions and millions of years. So
Yeah, but what about this secondary brain in the tail? I don't know where this came from. And they said they pointed out the tail and said it's got a secondary brain in the tail. There's proving that's
in the sacral vertebrae, there
yeah.
is the neural canal. So your neural canal, the neural canal in vertebrae is the kind of whole way your neural cord or your spinal cord runs through. And that is expanded and much wider in the vertebrae over the hip region, sacral vertebrae.
Oh, so it wasn't made up.
So it wasn't made up, and it has long been thought that that was maybe a collection of nerves or something there. We don't really know what occupied that space, but actually it's not unique to stegosaurs. It actually occurs in loads and loads of dinosaurs.
Okay, cool. All right, and so I can give that book a pass then, because I always thought, as I grew up, I thought that may be made up. Anyway, let's move on. So this idea then that you can think about endocast of fossils in the same way that you were just talking about with stegosaurs, or you do it with mammals, is try and think about the evolution of smell, how that's evolved over time. So the part of this paper is they start with this idea that the olfactory receptor genes, so these are the genes associated with smell, make up a substantial part of the mammalian genome. I looked that up. I thought, really, how much are we talking about here? So humans apparently have 400 genes associated with roughly, and that is of about 20,000 genes. So I'm not sure that's substantial. I mean, 400 genes is a lot, but of 20,000.
But primates have fewer, don't they? I only because I read this paper yesterday, and I'm sure you'll get to it. But it says that primates actually have a less good sense of smell than.
It varies quite a lot. It varies quite a lot within the mammals, which I thought was kind of interesting. I assumed it would vary quite a lot with the different kinds of groups you link to, but obviously it's much more closely linked to your ecology.
Yeah.
And actually, looking.
I was going to ask you, Rob. I mean, we haven't really said why sense of smell might be important, or why the evolution of sense of smell might be interesting. But actually, our discussion at the beginning kind of leads. It made me start to think about why smell might be important, and what things you smell for. And I guess detecting stuff that's decaying and not eating it, that might be like a really important thing, right? So that you don't get sick. Don't eat something that's decaying because it's going to make you sick. And then we talked about mammoth sex. Maybe pheromones and things like that, right? You can sniff a nice smelling other person or other badger. I was reading a paper about badgers this morning, so badgers are on my brain. I don't know.
Yeah, sex and death and eating. You need a good sense of smell. You need it to find your food, whether or not you're a meat eater or not, because you might want to find your fruits or your seeds or whatever, or avoid prey, find prey, avoid predators, find prey, and find your own members of your species. Figure out who smells good, figure out who's in heat, that sort of thing. So smell is important, but it's going to depend on your ecology quite a lot. And that's going to come up in this story later on about what you, and indeed, that's the sort of thing you might want to get a handle on if you're looking at fossil organisms. You might want to know a bit about the ecology. You might come up with some hypotheses about what a saber-toothed cat was doing or what a glyptodon was doing. You want to know what its ecology was and how did it, why is it like that? Why is it different from other organisms close by? You want to know a bit about what it was doing, and smell is an important part of that story, especially for mammals. So, it's very clearly a good starting point. So they point out that if you want to do this, you've got two ways to do this. One is to look at the size of your factory bulb. The other one is to look at the turbinates. Sorry, have you heard of turbinates before?
Yeah, I have. They're structures in the nose, and also I think they can they have maxillary turbinates in mammals as well, don't they? But they're in the nose, and they're like very convoluted passages which are used to warm air. So they're often thought to be associated with warm-blooded animals. So sometimes they're used as a kind of indicator of when endothermy evolved in mammals, I think, as well. Mm-hmm. I might be out of date on that.
Yeah, so you sometimes can get them fossilized because you've got these very thin, like convoluted bits of bone and cartilage inside your nose to sort of have the air go over all sorts of passages to keep it warm or cool or help with the smell. So that's kind of fun. Anyway, they start with this. Happens in dinosaurs too.
There are some sort of like turbinate-like structures and very convoluted nasal passages which are kind of similar in some dinosaurs. So ankylosaurs, some ankylosaurs have very, very convoluted nasal passages.
What does that mean that ankylosaurs were warm-blooded?
Been speculated, but it could be also something to do with sound that they were making, but we don't know.
Interesting. I wouldn't have ever thought ankylosaurs aren't the ones that I would immediately think of being warm-blooded. I see them as sort of slow lumbering ones, but what do I know? You're the dinosaur person. But anyway, that's cool. I did not know about these turbinates and ankylosaurs. That's a fun thing. So moving on to this, they've got a very clear hypothesis. Can we reconstruct the relative smellability and genetics of fossil organisms using endocast size? Right. I like that. That's a nice, very clear hypothesis. Boom. In we go. So the way they did this, they compiled 71 high-resolution head scans using micro CTs of mammals. So 71 is a lot. I mean, that is a lot of data to be working with. 40 of them, they scanned themselves. They got 40 heads in jars in ethanol, scanned them, took them to... I can't imagine how this would turn up. But I guess museums are used to this. Here we are.
It's like standard in the CT unit.
Yeah. Here's a wolf's head in a jar. Here's an aardvark in a jar. Just this decapitated head. Go ahead and scan that. But 31 they downloaded from Morphosource, which I think is kind of nice, right? So there's this, because there's more and more people do the scanning, they upload their data, they share their data, then anyone can come in and use it. I was chatting to Phil Cox about that. He was looking specifically at the ears, and he's been downloading loads and loads of ear data from all sorts of weird mammals, and it's all available and shared for people to use. That's cool. That's a bit of an open data story. So then they've got these massive scans. For the whales, they had a bit of difficulty because you don't really have a lot of CT scans of whales because you can't really fit them in a CT scanner.
You need like one of those ones that scan aircraft, don't you?
A bit more difficult to sit out with a decapitated whale and put it in the CT scanner. So they had to use this reconstructed endocast from a parasagittal sword specimen. So someone had just cut a brain and then they had to try and reconstruct it from that. Okay, so that's part one. And then part two, they extracted some genetic data from a previously published paper by someone else. Actually, I don't know if one of them was one of them the authors on this. I didn't actually get around to reading that paper. I was meant to go and read that paper.
I didn't read the genetics section because it's just lots of capital letters that don't mean anything to us. And funny order.
I was pretty sure you weren't going to. That's fine. But I'm feeling guilty because I didn't go and read the paper, the genetic paper that they cited where they harvested the data. But this is another example of open data, right?
Enough time and too many things.
No, I wasn't using this to tell you or I off. I was more using it as an example of how open science and sharing your data can be very powerful. That's what I was getting at.
Oh.
Yes. Not chastising you for not reading all the genetic acronyms. That's fine. That's okay. So they've got this large amount of genetic data where they've got the number of genes associated with olfaction and chemoreception and all these different taxa. And they introduce a lot of acronyms, which annoys me. Don't do that, people. If you can avoid introducing acronyms into your paper, please do that. Makes them more readable. So they've got these different genes. They've got the olfactory receptor genes, as in the ones associated with smell. They've got the ones that vomeral nasal genes, which I hadn't heard of before. These are ones associated with detecting pheromones. So we're back to pheromones again. So, some species pheromones are really important. I don't think humans use pheromones very much in comparison to other mammals, but some of them are very, very heavily dependent upon them. So, perhaps we should stop and explain what a pheromone is.
Which one? Rob, go ahead.
Let me just consult the Bodleian Library, Wikipedia.
Wikipedia.
Yeah. It's a chemical trait excreted by an organism which triggers a social response in the same species. So they're often some sort of, oily, chemically, highly volatile thing that they can detect. So there's a way of communicating between organisms, basically. I'm not explaining that very well, but it's not like a tasting something. It's a very different kind of airborne, often material. I'm blustering. I don't know. I should move on. I know roughly what a pheromone is, but I don't have a definition off the top of my head. Oh, God. How long can I get away with being the biologist and not being able to explain basic biological concepts to you? Shame.
Okay, I think it's fine. I mean, so I have a plum tree in my garden, and it gets all the plums get eaten by caterpillars. So we put a pheromone trap to capture the moths that lay the caterpillars, and it's a smelly thing that the moths really, really, really like. It smells like the female moths. And so all the male moths, it must be the other way around. It must smell like the male moths, so all the female moths fly towards it. Or
yes.
We trap all the moths, and then the caterpillars don't get laid on our plums, so we can eat them.
Perfect example.
That's I guess it's a thing that smells nice that attracts you to other people. And maybe humans do use it. We just use it in a more we just don't know that we're using it. Maybe it's more...
Yeah, I mean, there are experiments that you can do where they give subjects T-shirts to wear for a few days, and then They take the t-shirts off people, and then people in a different room smell them, and then so there are things going on with that people could do. But you just mentioned Matthew Cobb earlier. We should have perhaps talked to him about this episode because it's all about smell. But anyway, we didn't. So sorry, Matthew. So let's move on then. So we've got a big data set including all these morphologies of the endocasts. They calculated the endocast volume and the relative endocast volume, which is, try and weighing that against, I wasn't entirely sure whether they weighed that against the total brain size or the total body size, but, one of the two. So then they compared that onto all these phylogenies and all this genetic data. So what do we end up with? Well, first of all, the disparity and sort of diversity of olfactory bulb size is quite apparent, right? Some mammals, like an aardvark and a marsupial mole, have massive olfactory bulbs, and a dog, unsurprising, dogs have quite big ones, but the biggest one was an aardvark, which if you think about what an aardvark does, it doesn't really surprise me, right? Aardvarks with their little snuffle, that's basically what they spend all their entire time foraging around. And ones with basically no olfactory bulb at all was the river dolphin and a manatee, which is a very small one. So both aquatic ones. That's kind of interesting.
Well, it's kind of not surprising, is it? Because things, it's more difficult to smell underwater, I guess. You need volatiles, you need things in the air to be able to
Absolutely.
And you can't do that underwater. So yeah, so the things that have gone back to water have secondarily lost their ability to smell.
Not all of them though. So the some of the whales still do that because they do come up when they smell. When they come up and do the psh, there is a smell thing going on. But the river dolphins and the manatees, maybe it's something about the environment they live in where they don't have to worry about it so much. But yeah. So but they have got the relatively reduced one. What's a terrestrial animal that has a relatively small one? I'm just looking now. A tapir.
Wasn't an elephant? Didn't elephants have quite?
Yeah, elephants are weird though because they've got an exceedingly high amount of genetics. They have a very high number of smell genes, but they also have large brains and large body sizes, so it gets a bit weird with elephants. I think the other example is tapirs, which generally have a relatively small one. Oh, and a macaque, which is a kind of monkey type thing. Macaques have relatively small olfactory bulbs, and we're not massive actually. Now that we look at this in comparison to the rest of the mammals.
That was the primate thing. Yeah. I mean, it's interesting, isn't it? Because they've got a figure here which shows that kind of the skulls that they CT scanned, and then it shows the endocast and the olfactory bulb, and it's very clear how, I mean, mammals have lots of different shaped heads, right? If you think of our heads versus a dog's head, they're really, they've got a much longer snout, and it's sort of interesting how that impacts the shape of the brain. And I think they get into this a little bit, don't they, about how there's some constraints around how big your olfactory tract might be because of other things going on. But it's kind of interesting about how, which came first? What allowed what to change shape? Did the brain change shape and things like the olfactory? Is the reduction of a primate's olfactory lobe the result of having a short face and a small face and a smaller nose?
Yeah, it is interesting. Well, the dolphin's an extreme example of that because they've got extremely long snout.
Well, that's an unusual one, yeah, but that's about feeding, isn't it? That's about having that sort of elongate rostrum for catching fish, I guess. So that's yeah.
But when you look at this distribution of mammal skulls, you're right, they have a very different distribution. But I sometimes, when I think about this, think about how weird humans must look to other mammals, right? Because we've got our head is messed up. We don't have a snout or a nose or anything, but also our brain is enormous. Our skull, like if you look at a general mammal, we've got like this enormous swelling came out the top of our head. We must look like absolute freaks to other mammals. Like, what on earth has happened to that guy? Is what they must be thinking. Well, if they do think, because they didn't. Yes, it's my enormous swelling gargantuan brain on top of my tiny head. So let's have a look. Let's move on to some results. They have to do some complicated phylogenetics and phylogenetic comparative methods here because, you have to detract the evolutionary. If you want to pick up on the evolution of this thing, you have to take into account that animals more closely related to each other are going to be more similar to each other just because of the way that's how they evolve. So they're trying to detract the phylogeny, the underlying evolutionary pattern, and see how evolutionary smell goes up or down relative to that. So trying to get the baseline, and then they find a very tight correlation and a lovely series of graphs with nice figures on it, looking at there's a very tight correlation between the number of intact chemoreceptor genes and the relative volume of the endocast. So not the absolute volume, not how big your olfactory bulb is, but how big it is relative to either your brain or your body. So they find this nice tight correlation. So that's great. Now, I quite like that because that means that you can use that for fossils, and it means that you found a connection between genetics and morphology, which isn't easy to do. So in evolution, you often normally work on genetic data or morphological data, but it's rare that you find a direct connection between the two of them, just because it's very, very complicated relationship between the two of them.
Yeah, absolutely. And this is nice because, I mean, we've always assumed, and certainly we assume this in dinosaurs, as I said at the beginning, that things with big olfactory tracts must be better at smelling. But of course, though, there's actually demonstrating that here is it's helpful, isn't it? That is a good proxy. It's actually demonstrating that the proxy that we're using works.
Yeah, absolutely. So they do some statistics on this, and they demonstrate that, it's the relative one, not the absolute one, that's strongly correlated. And of those different categories of genes they've looked at, it's very much the olfactory genes that are the important ones, less so the pheromones, less so the taste receptors. So they looked at a whole bank of chemoreceptory ones, and it's mostly the olfactory ones, not the pheromones and taste, that are strongly correlated. So that's kind of fun. And as I say, a morphological genetic correlation. And now, so let's move on to what you can do with that. So you can start making predictions about fossils now. Before we move on to that, we have to talk about how there are sometimes some excursions from this, and this gets important later on. So they talk about some excursions from this relating to ecologies. The interesting one is manatees. So, they have this ecology where they're mostly in the water, but interestingly, and they don't use the air very much. Whereas there are some terrestrial fish that come out on land overnight and crawl around in the air, and they have evolved a lot of olfactory genes. So if you spend your time in the air, then you might end up evolving these olfactory genes, and if you don't, you might end up losing them. It's kind of interesting. So let's move on to the extinct organisms then. So they've got 5-6 extinct organisms which they plug into this to try and look at whether or not they were or were not relatively large in their use of smelling. So Smilodon is the saber-toothed cat. That's right slap bang in the middle, so that's looking very typical. One that they find quite high is the thylacine, the Tasmanian wolf, tiger, what do we call it? Tiger. So that one extinct relatively recently, and they indicate that one has a very high endocranial endocast volume for its olfactory bulbs. It might have been very well smelling. And the Glyptodon is this giant armadillo, and that one is quite low down on the smelling. This one might have reduced its amount of smelling style. And an interesting one is Protocetus. So Protocetus is a what do you call it? Protocetacean. It's of an early whale, a proto whale. So a stem group whale, should call it. So it's on the way to being a whale. It's one of these ones that's early in the transition of being a whale. But so it's not massively surprising that it has a relatively small endocranial volume, but of its olfactory system. But it has more. It hasn't got as little as the other cetaceans. So it hasn't yet completed its transition to losing its olfactory smelling ability. So it's consistent with it being a transitional whale, which is kind of fun.
Indeed. I actually thought that the Smilodon one was a little... I might have expected Smilodon, being a saber-toothed cat and being clearly a hypercarnivore, to maybe have been a little bit more higher up the line, as in it might have been better at smelling, but it's actually right in the middle, isn't it? It's not, as you said, it's not that much different from things like rabbits and stuff like that. It's not that far away from cats and dogs, though, cats and wolves, but yeah, I thought it might go a bit further up.
So that they compared Smilodon specifically with modern things that are like a canid, a dog, and they found that it had relatively reduced olfactory size comparison to others, which is interesting because, as you say, you might have expected to be quite sophisticated in its smelling abilities, but I guess the ones that they find with the elevated smelling abilities are not the carnivores, but the ones that spend their time grubbing about in the dirt and smelling out worms and things like Aardvark.
Moles and...
Moles. Oh yeah, moles are way off the scale. Aardvarks are off the scale. So if you're that sort of organism, if you're fossorial, if you're living in the dirt or whatever, then that's going to be an important one. Whereas Smilodon might be more typical, although dogs are quite high as well. So that's... But I guess the point is we're trying to get a handle on the ecology of extinct organisms. So it's indicating that it is possible. Although I'm going to give you two examples where it might be more of a struggle now. So we mentioned Glyptodon, the giant armadillo. So Glyptodon is weird. If you don't know what a Glyptodon is, look it up. It's an armadillo the size of a small car. Uh, so that's already very alarming. Lived in South America, but it has a relatively reduced smell size for what they might have expected otherwise. But they also said that that might be because it was relying instead on the turbinates, those small bones. There's a slight balance between the two there. So this is where it gets a bit difficult here in how much of a correlation can you have that it is reliable. Similarly with the thylacine, they found that the thylacine, this Tasmanian tiger, has a high value, but when they looked at the genes, they found that maybe the genes in that case, because the Tasmanian tiger is relatively recent, you can actually reconstruct some of the genes, although they're not complete, and they found that there was potentially a discrepancy there. So this is the slight difficulty that arises here. Even though they found a strong correlation between the genetics and the morphology, is it strong enough that you could make predictions? And the answer is possibly not reliably. Even though the correlation is strong, is it strong enough to make predictions from the extinct organisms? Maybe, maybe not. So that's a fun story, but it does have that caveat. But I guess that's kind of true for all studies like this. What do you think?
It is, but I do think that you can make, I mean, you can't be specific. I wouldn't want to be reading off the number of intact chemoreceptor genes that there were from graph for an extinct animal. But I think it gives you a ballpark of where these things are, right? It clearly, Protocetus, as you said, is an interesting one because it's an early whale. And we know that things that are living in marine environments are tending to reduce their chemoreceptors and their ability to smell. And Protocetus doesn't have as few chemoreceptors or as living whales, but it is on its way. It's lower than other mammals. So I think you can make some general statements, although you probably want to err on the side of caution and not make specifics.
Yeah, I was going to reach a similar sort of conclusion, I guess, was that, first of all, I think this is really interesting in the sense that you can take all this massive amount of data and reconstruct a trend and make a connection between genetics and morphology, which is unusual. Normally, that's very hard. But because of the fact that the correlation is significant and it's strong, but it's not enough. So you wouldn't ever be able to do it on a one-to-one basis. You couldn't ever really say, I've got this fossil organism, and I think that because of its endocast, of its olfactory bulb, it was doing X, Y, Z. But you might be able to pick up on trends. Like if you included a lot of fossil data alongside a lot of modern data, that very much similar to what you were saying, I guess, is you could pick up on those things. So I might not be able to look at Protocetus on its own, but I might be able to look at Protocetus alongside Bacillodon, alongside all these other whales, fossils and modern, and then pick up an evolutionary trend through time, which is perhaps more powerful and interesting.
It would be great to know how reptiles fit these similar graphs as well, actually, and whether this is a general pattern that applies to all animals, whether these regressions are different for different types of animals, or whether a mammal's good representatives of everything. Can we apply dinosaurs to the mammal graph? Can we apply birds to the mammal graph? That would be an interesting thing to see.
Yeah, I think that would be cool. I mean, I guess I'm now looking at reptiles' olfactory bulb pictures. It looks like their olfactory bulb is even more distinct from the rest of their brain than in mammals. It looks like it's quite discrete, so it might be possible.
Yeah, it is, and I guess that some of that might be because maybe the rest of the brain is smaller or rather different from mammalian brains. So it could be that you have these very distinct olfactory bulbs. I know T-Rex has very big olfactory bulbs, thanks to Steve Brasati's book. So there are brains in dinosaurs where we have very distinct, olfactory bulbs and olfactory passages that can be traced.
So that's possible. There's one possibility there. I guess the difficulty is that you could definitely do this with reptiles, I guess, but birds might be a bit different, and they're perhaps the best model. And there's quite a big gap between.
Do we know anything about bird smell? Do birds use their sense of smell a lot? I know nothing about that.
I'm going quiet because I am trying to fill the gap in. Before I admit that I know very little about the way birds smell.
I think that's okay. I think just because you're a biologist, you don't have to know about the way that everything works, Rob. I mean, I'm gonna ask you on the assumption that you do, but it's okay if you don't.
No, I'm afraid I don't know much about bird smell, but it should be possible, right? So one of the things that they did remark about, so if you're out there thinking, I want to go do this study with reptiles and birds, one of the take-home messages is that you have to rely a lot on the open data, right? Mammals have an awful lot of those skulls uploaded and available to share. I don't know if the same data is available for birds and reptiles. It probably is, but not maybe to the same degree. Mammals are perhaps one of the most well scanned and uploaded data sets. The other thing is that the authors talk about how difficult it was in some cases to demark specifically where the olfactory bulb is, because when you've got this data, even when you have a really nice scan, there's still some level of subjectivity of drawing a line between where one part of the brain begins, another part of the brain ends, especially whether it's an endocast from a fossil. So that's something to take. And they found that the different people measuring the same thing came to different conclusions. So you have to be a bit careful about that. Again, back to the same old classic morphological problem. How do you measure morphology? It gets a bit difficult. So that's the story of mammal smell, I guess. We're a bit closer to understanding how smells evolved and how mammals would detect them. And you can do that if you do a big data set, but maybe not on one or two of them, but they're on their own.
Yeah, I think there's a good take home here, which is that we can use the morphology of endocasts to get general patterns about the sense of smell in extinct animals, which could be really helpful in helping us understand their ecology.
Yeah, that's a nice sum. Okay, well, thanks, Susie. I'm going to pass over to you to think about something else for us to talk about next time, and I'll see all our listeners for our next episode. The Fossil Files was recorded by Robert Sansom and Susanna Maidman.
We want to hear what you think. You can let us know your thoughts on the papers that we have discussed, and you can make suggestions for papers for us to talk about on our social media channels.
You can find our email address and links to our Instagram, Blue Sky and YouTube channels on our website, fossils.libsyn.com.